If you haven't been here before, thank you for coming today. There are some unfamiliar faces, so thanks so much for stopping by. I'm really excited about this program because I got these books out for Allison to look through. I introduced my son to this book a long time ago, and it's by Eric Sloan, Diary of an Early American Boy. And if you read his books, the illustrations are wonderful. He was an artist, but he wrote about early American life. And he talks about the American chestnut and how important it was. And I was just intrigued. So when Allison said yes, I was just so happy that she could come. So before we get started, please turn off your cell phones, or at least the ringer, so that they don't go off during the program. I've moved all our phones into the other room. And I guess that's it. Allison, thank you so much for coming.
Oh, thank you for having me. So there's plenty of seating, by the way. All right, so I am Dr. Allison Oakes. I've come down the long, arduous trip down 81 in full fall color. It was lovely. From Syracuse, New York. I'm with the College of Environmental Science and Forestry. We were a state school, then we were attached to Syracuse University. Now we're a state school again. But our specialty is in sort of environmental topics, and a lot of, we have just straight science, and then we have environmental studies. We have a paper and bioprocess engineering team, a chemistry department, so we're a great little, if anybody wants to push their grandchildren towards environmental type things, we're a great school. It's nice and small, and the professor to student ratio is quite high still, unlike a lot of schools today. So. I've been part of the American Chestnut Research and Restoration Project for about 13 years now. So I started as an undergrad who needed a job, and I washed test tubes for this lab and discovered they were doing some really interesting research. So changed my major, became part of the group, and worked for them for a long time. Got my undergrad degree in 2009 and my PhD in 2015. And now I'm a postdoc, and they can't seem to get rid of me, or at least they keep asking me to work for them, so that's good. Full-time research scientist with the project, and our main focus has been on the American chestnut. Okay, so it's a hardwood tree that grows relatively rapidly for a hardwood. Okay, this is one of our seedlings here. When they reach the canopy and they get full light, they flower, and they have these huge top of the trees in full bloom right around mid-June. And one of the things we've been doing to find isolated American chestnuts is to fly a drone over the top of the forest and look for these like giant puffy firework, almost like flowers. We like to say that they, explode on the 4th of July, right? And they have a very strong scent to them, which is once you smell it, you will always remember it. And then these guys, the American chestnut is one of the biggest, largest of the Castanea trees. It was called here's a 6-foot lumberjack by comparison. It was called the redwood of the East. Wow. They got absolutely huge. And the wood is fantastic for building, and the nuts are fantastic for eating. So the nuts are very nutritious, high in protein and fat, and the wood is rot resistant. You can see a lot of the barns that have just lasted forever and don't seem to fall down are actually made of chestnut, since they're such a prevalent wood. It's right up there with cedar in terms of longevity, so if you want to build a deck with it and have that deck last for 100, 150 years, chestnut will do that. It's gluten-free. It's it's a great baking flour. I highly recommend. I make a dark chocolate pumpkin cookie with it, and you replace about a quarter of the flour with chestnut flour, and it gives this great nuttiness and flavor to it. It makes excellent beer. Our greenhouse manager, Jeff Zarnowski, his wife, Dawn Zarnowski, I think runs the Cortland Brewing Company, and they've been experimenting with hazelnut and chestnut beers. Very, very good, highly approved. And then throughout the ecology of our forests, many things ate chestnuts. So from bears to deer to turkeys, passenger pigeons, even the chestnuts, the American ones are small enough. So here's kind of a large one and a small one that many birds actually can eat these guys, as opposed to the larger Chinese chestnuts. And there's a fish in here for some reason. Well, that's because every fall, the leaves from all of our deciduous trees are falling into the streams, right? They're falling, and the tannins are leaching into the water, and the fish and the invertebrates are nibbling on the leaves, and it turns out that almost all the species in our area have co-evolved with the American chestnut for millions of years. And so if we give invertebrates or tadpoles chestnut leaves, they grow faster and they are healthier than stuff that's grown on like maple or beech or oak, because chestnut was the predominant tree for such a long time. Had a massive range. I gave a talk like this up in Maine. They were like, that's not the range of the American chestnut in Maine. We found it all the way. I'm like, I'm sorry, this is from, Pallet 2007. I'll let him know that the range map extends up into northern Maine. Okay, but it goes all the way down into the highlands of Mississippi, Arkansas, and even I have a friend who works in Montreal, and she's been studying the southern Ontario lineage of chestnuts, and so it covered just the whole range of the Appalachians, and definitely up in our area as well. And if not the Catskills proper, definitely certain areas within the Catskills did have a large amount of chestnut population. So chestnut blight is a fungus. It enters the chestnut through a crack in the bark, and unlike the Asian species with which it co-evolved, it was brought over on Chinese chestnuts, it's fine there, but on American chestnuts, it gets in through a little tiny hole in the bark, and then it just spreads and spreads and spreads and spreads until it's all the way around the tree and it girdles it. So I've got a couple of examples here. So like in the crotch of the limbs here, it can get in once the bark is, the tree is big enough to have cracks in the bark, so usually around, we've seen it as early as 5 years, but usually more like 8 or 10 is when you start seeing these infections. Everything above the top of that infection dies, and then usually the chestnut can re-sprout from the bottom, which is fine, but you've lost your main tree, right? So you have little stump sprouts coming up. They're not getting old enough to reproduce. They can't get to the canopy. They can't, because every time they get tall enough, they get killed back. And so this occurred in the early 1900s, about 1904, 1908, and it was just devastating. If you can imagine, almost 25% of the trees in your forests dying of a pathogen that you've never seen before. Again, luckily they do sprout from the stumps, so people are finding, oh, I found a chestnut in the woods. Yes, they're still there. They are there, but they're not breeding. And the key to evolving resistance over time would be to have all the chestnuts breeding and making new offspring, and the offspring having some kind of new genetic makeup that would allow them to resist the disease, but these guys can't get big enough to breed. So there's no change. They're just the same trees sprouting over again. So it took about 50 years from, they think it was imported in the 1890s. It took about 50 years for it to just spread from the epicenter in New York City, Long Island area, out, further. There are other chestnut species, the Allegheny chinquapin, that's sort of along the coastal plains. It's a little bushy bush that also produces nuts and everything. And then we have the Ozark chinquapin out here in the Ozark Mountains, but that got hit in like the 1950s, 1960s. So there's still people alive that remember going out to the woods and collecting all the chestnuts, like we hear from, the generation previous, people that were alive more in the 1890s, 1910s remember it here, but it's really neat that they still have a lot of the history of chestnut use in the Ozarks as opposed to on the eastern seaboard. But eventually the blight came and hit them as well. Now the reason we can't go back out and just plant trees is because Cryphonectria parasitica, this fungus, survives on oak trees. Anywhere there's an oak tree, there's still some chestnut blight hanging around. You plant a chestnut, looks fine for about 10 years, and then the blight hits because we have this reservoir of fungus just hanging out. So here's a picture from 1912 of this is the fluffy flower tops, and almost looks like frosting on top of the trees, and just the how many trees were in these woods at that time is sort of mind-blowing. So, and here's in New York and Long Island what that forest would have looked like in terms of just death of trees. So they tried as best they could. They cut down fire breaks. They tried to prevent the fungus, but like I said, it survives on oak trees, so it would just kind of, and it flies on the wind, very tiny spores, so it would just fly right over all the fire breaks. So they actually might have done a lot more harm than good by cutting down all these healthy trees, because you really, brought down the genetic diversity of the population. So they are around. They do survive, but every time they come up, they lose stored carbon in their roots. There's only so many times that they can sprout from the root system until the whole stump just dies. So it's sort of a race against time to catch these guys. They do sprout. We had one grow 7 feet in one season that we cut back. They are very fast growers. but every time they grow, they eventually roll back. It's kind of a Sisyphus battle where they just can't seem to keep going. So people have been trying breeding and lots of different options for a long time. What about all the chestnuts you see in the market? You can go to Lowe's and get a Dunstan chestnut for 25 bucks or something. Well, it's a hybrid. Dunstan chestnut is not a particularly, it's not a clonal line. The nuts are very big. They're not quite what wildlife are used to, so especially if you want birds and fowl and stuff, they're too big for birds to eat as opposed to regular Americans. And they are kind of a giant mutt, which is super great if you want nuts for deer and bears, or you just want nuts to eat. Sure, fine, they're a great chestnut. American, Chinese, Japanese, European, Seguin, they're a giant mix. It's a seed lot. It's not an actual like patented clonal variety, but they do grow. If that's all you want are chestnuts, then any of these are perfectly great. But they're not going to do this. They're not going to get this big. They're not going to be a canopy tree that really is part of the native ecosystem with all these different traits and genes from chestnuts around the world. So for restoration projects, we really want something as close to the original American as we can get. So radiation mutational breeding, because in the 60s and 70s, why not? X-rays and radiation is cool. They basically just doused chestnut seeds with radiation and planted them to see if any resistance would happen. It didn't, because that's not how it works. Like a real mutation is not Spider-Man with superpowers appearing. So we have those trees, but none of them really started to show resistance. So the American Chestnut Foundation, which is every state that had chestnuts basically has a chapter. They started this Burnham program when they thought back in the 50s there must be about 3 genes for resistance. What we can do is we can breed American with Chinese, and then we'll breed it back to American, and then we'll select for trees that are most resistant to the blight. And so they did this, they backcrossed, and the goal was to get a tree that was 15/16 mostly American with a little bit of Chinese, just enough Chinese to get those blight resistance traits. Well, it was a good plan, the problem being that back in the 1950s we didn't have the DNA sequencing technology that we do today, and further genetic analysis has found there's probably about 9 to 20 separate genes that all work together on, I think, 9 out of the 12 chromosomes that all have to be present in order to get that level of blight resistance. So as soon as you breed it with a not-resistant tree, you have lost half of the blight resistance. So you're going to dilute out the resistance. They didn't know that when they started. They had a very good plan. It totally would have worked if it had been 3 genes, but unfortunately it was a lot more than that. So they also had some contamination in their orchards with the Chinese chestnut accidentally contaminating stuff down the line. But so they're going back to the drawing board, reviewing what they have, and they're really right now, the TACF, and we've been talking to them too about how to do marker-assisted breeding or continue with this project. But so they do have resistant trees, but again, they're more Chinese than they thought, and they're not going to have the genetic resistance isn't going to be as strong because there's so many different genes playing into this. Okay? So in the 1980s, the New York State chapter of American Chestnut Foundation came to the College of Environmental Science and Forestry, and they said to Dr. Powell and Dr. Maynard, we've been hearing about this genetically modifying crops thing. This sounds kind of neat. Is there any way that we could just find a gene that would give resistance, full resistance to the American chestnut? We could put that in. So the thing here is that chestnut has about 40,000 genes. Okay, so if you have a 1/16 Chinese chestnut genes, you have removed 2,500. This is a unit of information, coding proteins and all that. You've removed a lot of information from the American and replaced it with the Chinese information, which could be fine. So, but think about it as a book, just a regular, 200-page, paperback book. You have removed 10 pages of text and replaced them with 10 pages of a book text from a different book. Okay, so about 2,500 words. So it could be a big change. It could be not that important. It could have been like the preface or the index or something. You don't really know. Whereas with genetic engineering, the level of change is adding 2 words to a book. It's minuscule. And we know what the words are exactly that we're putting in. So in this case, we could add, it was very exciting at that season to roam the then boundless, blight-resistant chestnut woods of Lincoln. Just on a scale of change, genetic engineering makes such tiny changes And other thing it does is it doesn't remove any of the material or any of the information that was there. It just sort of inserts a piece in. Okay, so are you going to play for me? Excellent. So we looked at the chestnut fungus here. I don't think it's playing. There's the play button. There we go.
Oh, the volume might be way down.
That's okay. It has overly dramatic music that I don't mind missing. Okay, so my deciding not to play. What would happen here is we can use an apple to simulate the growth of the fungus. Surprise, somebody found that out way back when. And the one that produces a small amount of acid doesn't hurt the surrounding tissue as much as this strain that produces a lot of acid. Okay, so oxalate is oxalic acid. So you have a little bit here and a lot there, and what you would see if my video is working is that the one that exudes a lot of acid kills the tissue much faster, the fungus expands, and we found that the oxalic acid is the main weapon of the fungus that it uses to attack the tree. So what we found is a gene from wheat called oxalate oxidase, okay, and what it does is it just breaks down that acid. This, it's not a pesticide, it doesn't hurt the fungus at all. It reduces, so then the fungus has less of a chance of developing resistance to this particular gene. And then it also, since it doesn't kill the fungus happily lives on the tissue but can't expand further. It's sort of like a nice little containment strategy. And this gene though, I mean, people, switching genes between species, is that really something that's conducive? Many plants have this gene. So if you ate a bagel for breakfast, you ate this enzyme here. If you had rice cereal instead of wheat cereal, you're eating wheat, that's cool. Rice has this gene too. All of these plants have a variant of this particular enzyme because many fungi attack plants with oxalic acid. So many plants have evolved resistance to this acid. So I'm sure there's others, but we can only go with what's been published on the web so far and search our gene databases. Okay? The other nice thing about using a one gene system is that there's a very simple test to show whether or not the gene is present. You can take a little bit of tissue and put it in a solution that's rather cheap and easy to make, and if the gene is present, it will stain a nice dark ring. So here, you can grab these if you want, pass them around. So if anybody's like, I don't want these on my property, all right, come down, use a little hole punch, bink, put them in the solution, wait about an hour, and if it has the transgene, it will stain a nice dark color. And you can say, okay, it's gone. So we don't need expensive PCR or genetic testing to determine if the tree is transgenic. If anybody doesn't want it, it's easy to find. And again, if you do want it and you want to know that the tree is, you have a bunch of seeds, you grow them all up and you want to know which ones have the gene, it's very easy to test. Okay. You just literally cut a little piece out, add the solution, and then you have, if it's not transgenic, it won't stain. If it is transgenic, there's a chemical in this that will react with the hydrogen peroxide produced by the reaction in order to show a dark stain. How do you get the gene in? Very simple. Just follow these steps. There's only 3 checkpoints at which we check like quality control wise, but the scientists just toss a gene right in. It's really difficult and time consuming and expensive. So we get what gene we want. In this case, we want the oxalate oxidase, and we use a bacteria found in nature called Agrobacterium. It lives in the soil, and the way it helps itself is by inserting some of its own genes into plants. And usually that's something we don't really want, which makes crown gall. So what scientists said though is, what if I took this piece that bacteria is inserting into its host, and what if I put the gene I wanted in there instead? And lo and behold, that works, and you can engineer plants using this bacteria. Agrobacterium has inserted its own genes into about 2,000 different plant species. Does anybody like sweet potato pie? Mm-hmm. Naturally genetic engineered by Agrobacterium. It has bacterial DNA in it. It seems to be fine and delicious and not a problem to anybody at all. So, and monarch butterflies also, not by Agrobacterium, but they have genes that were inserted by a virus about, I think 50,000 years ago. So the monarch butterfly is also a genetically modified organism. which makes it hilarious that it's the flagship of the non-GMO project. Okay. All right, so first you take these little embryos, you open up the nuts of the chestnut when they're very, very tiny, and you take the little embryos out of the nuts. This has about a 1 in 1,000 success rate. Okay, so there they are. You open up the nut, you get these little tiny, so this one's becoming the dominant seed there, but these guys you can harvest and put onto plates of plant media and help them grow. They really look like little boogers. They're pretty gross to work with. But you have all of these little tiny plant embryos growing, a culture of those, and what we do is mix them with that Agrobacterium that has our gene of interest. We swish them together, then we select for the ones that have been transformed, and at the end of this, you have a clump of chestnut embryos that contain the gene of interest. In our case, this is oxalate oxidase, and anything grown from these embryos will have that gene. So now we go to germinating them. We put them on a different set of hormones at a different media, and eventually you get one little tiny shoot right there, and that grows up into a little mini chestnut culture. I didn't have any non-transgenic ones, or I would have brought some down, but I can't move those without a permit. And then you take these tiny little shoots, and just like a tomato cutting, or if anybody's taken rose cuttings or anything like that, you put it, dip it in some rooting hormone, put it in a nice wet spot, and then put it in some soil, let them grow, and then suddenly we start having nice clonally propagated plants here, all of which have the gene of interest. Once they're big enough and sturdy enough, they go to the greenhouse. So this is our greenhouse full of transgenic chestnut trees here. And then once they're big enough, we do some stem assays. What we do is we make a little cut in the stem of the tree, rub some fungus on there, and wait and see if it dies. And so here's one of our most recent ones where we did this to pure American chestnut and they all died, but you can see they're starting to sprout from the base, just like a chestnut out in the woods. The transgenic ones we tested, nothing. They sat there, they produced their enzyme, they destroyed that toxic acid, and they kept growing. And our Chinese chestnut, actually 3 of them died, and 3 of them survived. So we actually had better blight resistance than in the naturally resistant Chinese chestnut, which is very, very exciting. Is it heritable? Can this transmit to its children? Because we don't want to keep putting chestnuts in the forest and having them die. And yes, it is. So here's the transgenic. These are siblings. This was one that inherited the gene. And so you see we made a little cut, put the fungus in, it made a little canker. You can see a little bit of orange in the fungus around there. But it lived. It walled it off. It's able to live, both the tree and the fungus. Here's its sibling that didn't get the gene. This is what most American chestnut look like after inoculating. Okay. So it's a 50-50 chance if the children get the gene, but what we're intending to do, because we don't want to plant a clone, just clone orchards, that's a terrible idea, right? Like if something else comes in, another parasite is going to die. We want all the diversity that's out there in the surviving American chestnuts, the ones that have been holding on to life. And so the idea is, this is what the New York chapter president, Nichols, is pushing for right now, is making lots and lots of mother tree American chestnuts. If people go out and plant just wild type chestnuts, we know they're going to die, but in about 5 to 8 years, they'll start having flowers, they'll start having female flowers, and then we could cross our transgenic pollen to all those regionally adapted, diverse trees, and then half of the offspring is going to inherit that gene. Okay, and we're going to get different complements of genes from mom and dad. Okay, and then we're capturing the diversity from those locally adapted trees and keep outcrossing, and keep getting as diverse as we can, because nobody wants a clonal tree orchard. Nobody thinks that's a good idea. So that's what we're working on now, is this mother tree plan. We're regulated by the federal government because we used Agrobacterium to insert the gene, and that's technically a plant pest, even though none of it remains in the chestnut. So we're currently going through a whole deregulation process. We'd like to be able to let them flower, see how far the pollen travels, let them cross in between each other, but currently every flower on this tree has to be bagged. So you can understand how much fun, either cut off or bag, you can understand how much fun that is to try and get a 30-foot tall tree and cut off or bag every flower. So eventually when they get too tall, we literally just have to cut them down and let them sprout and wait a few years. It's very tricky to do. So we use a bucket truck to go up there. In these cases, these are the female flowers, and we're going and pollinating select female flowers on a wild American chestnut. So these are some of our wild trees that we have in our Lafayette Road planting. And you get way up there and we brush the pollen on just like a little bee. And then eventually, once they have grown into nuts, we just had this harvest a few weeks ago. We cut all the bags off, bring them into the lab, and then each nut gets tagged with a certain number and weighed and put in a, and then but we keep them over the winter. So if you want to store chestnuts to plant in the spring, you get some moist peat moss, just a little moist peat moss, tuck the chestnuts in and put it in your fridge and forget about it until about March, okay? Then we germinate them in our greenhouses and we plant them in our fields and wait and see how well they grow, which if the woodchucks don't eat them, they do very well. Oh my gosh, woodchucks are our bane, I swear. And so this is a seedling that doesn't have the gene for oxalate oxidase and has been growing very happily. I think it's about 12 feet now. It got a really good growing season this year. So, like I said, we're still regulated because we use this type of technology to make these trees. So we're currently working with and very positively feedback from all of them, USDA, FDA, and the EPA. And so it's going to probably take, we're hoping more like 2 to 3 years. It's very expensive, so luckily we have help from some of the federal agencies and other groups, but it's a very expensive process and people just cannot go plant stuff willy-nilly. Am I about 2 minutes in? So, okay. So one of the biggest things they want to know, which is what we've spent the past about 5 years really focusing on, is how safe are these trees? Are there going to be any environmental effects? Are there going to be off-target effects? So we've looked at mycorrhizae, which is the soil fungus that makes symbiosis with trees. And leaf decomposition, growth habits, do stream invertebrates have problems with the leaves, herbaceous understory, insect leaf feeding, leaf litter, oxo, and things like looking at tadpoles and bees are some of the newest stuff that we've done. But basically there is a lot of safety testing here. So one of them being does, like the leaves, right, that are falling from the trees, it has this anti-acid enzyme. Is that going to affect any other seeds that are trying to germinate in the leaf litter? So we set up some leaf litter trays with different types of chestnuts and different types of seeds, incubated them, and then germinated, and saw that there's a lot of variation between, so these are different, these colors here are the different plants that we looked at. So does maple germinate or pine? And we had some other understory plants and found that between not having any leaves, having transgenic, a non-transgenic, and a non-transgenic, the most significant difference between maples was two randomly not related American chestnuts that didn't have the trans gene. And here are the two that do. They weren't significantly different. So we found some interesting stuff here. So for example, this transgenic and this non-transgenic were different, but this was the same as this non-transgenic. So we're seeing a lot of difference just in the natural variation of how different chestnuts behave as opposed to whether or not they have a transgene or not. So I can definitely go over these more if you like, but running out of time. So what about mycorrhizal symbiosis, the beneficial root fungus that helps the tree gain nutrients? Is there any difference there? So we had a bunch of soil from the forest, mixed that in with some potting mix and germinated a whole bunch of chestnuts in it and saw what mycorrhizae formed. Some poor grad student got to look at about 5,000 slide views and count how many colonized root tips there were and then tried to morphotype them and what species it was. What it really came down to is where we got our soil was significant. The soil from the woods had more mycorrhizae than the soil from the fields and such. And then we had, what the big difference here was, here's our wild type and our transgenic, right about the same. The different colonization here was on hybrids. Chinese chestnut and hybrids had lower mycorrhizal colonization. Makes sense. They have a different genetic complement. Chinese chestnut is from a different continent, so it's not going to interact as well with the local mycorrhizae. So, okay. Thank you for the boxes. And then we looked at tadpoles. Oh my goodness, this was the cutest experiment ever. And some poor undergrad had to change the water in every one of these jars once a week for 4 months or something. So science, it's fun. And one of the things we looked at was Gossamer stage. Gossamer stage is how tadpoles change in development over time when you start seeing the bulges where the legs are going to pop out and such. And so we found that there really wasn't a difference between the transgenic chestnut and the non-transgenic, and we have the hybrid, the Chinese, the sugar maple, and the American beech. They all seem to be right about the same, not a whole lot of difference. That was good, we're doing okay. However, if we looked at ones that didn't receive a food supplement and looked at how long it took them to, for them to reach a certain Gossamer stage, they did best on the transgenic chestnut and the regular chestnut. But if you give them hybrid chestnut or Chinese or sugar maple or beech, they grow slower. They don't have, for some reason, it makes sense. Wood frogs co-evolved with chestnut over thousands of years, so that's their preferred diet. It's been missing for 100 years, but that's not long enough to really make a huge change. But they don't grow, so they're surviving. They're doing okay on these other trees, but they really prefer that chestnut. And then finally, what about bees? Pollinators are a hot topic. We would hate to have anything severely impact a pollinator, especially in the native ones. So what happens if we give some bees? And here we've collected some chestnut pollen. We have a beehive. We actually got chestnut honey this year, which is really neat. We're hoping to ramp that up because it's got an amazing flavor. It's kind of a blend. It's like a chestnut wildflower blend because we didn't collect right at the end of chestnut pollen season, but it is really good. And we made these mini hives with little tiny bees. A very nice researcher from Cornell loaned us these mini hives. And if you get 5 or 6 bees, they start to build little, oh, they're there. They're building little egg cells, and they're actually depositing larvae, and they mimic what a natural hive would be, because we do not have the money to make 50 hives right now, so we had to use mini ones. And we looked at bee survival over time, so this is all the bees are alive, and then eventually bees only live about 40 days or so, 40 to 60 days depending on the thing. And so we had no oxalate oxidase in the pollen. We had high amounts of the enzyme in the pollen and a standard rate in the pollen, and we really saw that whatever pollen treatment they got, it all seemed to be about the same survival curve. Now, for comparison, we bought 3 different hives of bees for our replicates, and there was way more difference we bought hive A, B, and C, just 3 of them, and split them between the treatments. There's way more of a difference from what hive they came from than from what pollen they ate. So that's also promising. So we have had a lot of help over the years, lots and lots of funding from the American Chestnut Foundation, specifically the New York State chapter. And if you're interested in that, I recommend this little flyer is where you can come see like our work. It's got our website here. And this has just been hundreds and hundreds of people working on. Here's a little bit, this was my PhD basically, was how to get these things to root so that we could put them in the greenhouse and get them out in the field. And yeah, it's been a wonderful time working with them. Thank you guys for having me here to talk about this, and I'm totally open for questions. So, yes?
A few years ago, the Soil and Water Conservation District was selling Chinese chestnuts.
Yep.
And I planted 6 of them. Mm-hmm. And there's 2 that are alive but dying. Okay. And they're doing exactly what you said the American chestnuts did. In other words, they die. Yep. They're about 20 feet tall. Mm-hmm. And they die, and then lots of sprouts come up around the bottom.
Yeah.
Am I doing anybody any favors by not killing those things off? I mean, it'd be better to
Depends if you want, just orchard chestnuts, for consumption or for wildlife, then they're fine. So.
The other thing is, the nut is inside this.
Oh yeah.
Obnoxious cone. Is that true of the American chestnut as well?
Here,
those things are horrible.
Oh yeah, they're
terrible. I mean, they're not. I thought they were soft when they first started.
When they're green and they fall off, we have this very scientific method of like ripping them open with your shoes because nobody wants to actually touch them with gloves. But if you leave them long enough, if you just let them dry for a couple days, they'll open up like this, and then the nuts will just fall out. So that's our other thing, is either stomp on them or just let them, and as they dry out, they'll crack.
They're not good for a yard tree. They may be good for timber, but for
like at the back of your yard, if you want some deer coming, to the back of the yard, then sure, but you don't want them right by your driveway.
Yeah. I have another question along that same line. We're starting to hear now a lot of places that are selling American chestnut trees.
Right. American chestnut trees.
What are they?
Depends. It's not a named variety, so they could literally sell you anything and call it an American chestnut.
None of these things, these darlings or anything like that, none of that's available to the public yet, is it?
None of our transgenic trees are available. If you see anything labeled as a blight-resistant American, it's probably one of the crosses with American and Chinese. And unless they give you a genealogy or explain to you like, oh, this is how we got this tree, you're not going to know. It's sort of a buyer beware market because there aren't any named varieties that are protected by like the Plant Patent Act or anything like that. So, I mean, there's, like I said, if you want chestnuts to eat or chestnuts for wildlife, any of those will do just fine.
But we're not ready to start growing American chestnut trees for timber. Yeah.
Not yet, not until these guys start getting out there and breeding. Yeah. So, but like I said, that's where you could plant the mother trees. And if you had mother trees that could at least incubate the seeds, the offspring of the transgenic trees, then you could get a whole basket of nuts and plant those out. And half of them are going to have that gene and be able to live their full
But
life
here,
cycle.
get back to the Chinese chestnut. You don't want those Chinese chestnuts to be the mother tree. Right?
Not if you want timber type.
Right.
There are timber type Chinese chestnuts, Seguini and Henry E, but those haven't really been brought over to the US or worked with. That's one thing that the American Chestnut Foundation is looking at, but you're looking at starting a 60-year breeding program now if you go back and start incorporating a new species in your breeding program. So, yes.
How old were the trees in the 1904 photograph, you think?
Oh, those huge guys, hundreds.
Hundreds of years old.
I mean, I think they found like
So
450.
were they used for, non-structural timber for decorative timber in homes 150 years ago?
Structural.
I thought they used hemlock for the structural and
Oh,
used
okay.
the hemlock.
Okay.
Hemlock was the structural stuff and the
Okay.
chestnut was the decorative
finishing.
stuff. Yeah.
A lot of the old barns have
Mhm.
chestnut timbers inside.
Mhm.
Yes.
The
I think
hemlock,
it's
As opposed
what
to
was
be-
available.
the
yeah.
hemlock wasn't the hemlock on the outside, on the siding and things like that? I don't know.
Not a woodworker.
No, I mean, it's basically all the structural stuff in any of the buildings
Okay.
down on Main
Oh,
Street.
Okay. Also depends, was there a nice big hemlock forest and that's what was used to build everything as opposed to was there, people did build with what was available at the time. So I don't know if it was trucked in or moved around. That would be a question for the historians present. Yes?
You spoke of the fungus coming in, New York, Long Island,
Mhm.
New England, from where?
Asia. They started shipping in decorative trees. So somebody brought over a whole load of Chinese and Japanese chestnuts from Asia that, of course, had the fungus on them when they brought them in. That's the story of almost every plant pathogen we have here. Somebody wanted a pretty tree, and over it came. So that's the same thing with the elms. People brought over decorative elms from Asia and Europe, and lo and behold, they had Dutch elm disease on them. And American elm couldn't handle it, and poof.
So is there any living American chestnut growing outside America someplace?
Out west, people took the chestnuts with them when they went out. And so there's lots of, you can see in Nebraska, there were some chestnuts out there. There's some chestnuts on the west coast. There's actually some huge ones out in Washington and Oregon. But the problem is it's a limited diversity. There's just a couple of trees out there that are doing well. And so we would like that diversity, but we're also worried if we go out there and collect anything or do anything with the trees, we might bring the fungus to those areas. So we literally don't travel to where, our research group anyway, don't travel to where there are healthy chestnuts.
So there
in
is
general.
healthy chestnuts in the western
Yes.
states
Yeah.
in very large quantity?
Like here and there, like somebody would plant 2 in front of their house. There's not really American chestnut orchards. And they're trying. They're increasing production. They're seeing that chestnut is a good nut crop. But overall, and the other thing with chestnuts is they can't self-pollinate. If you have one tree, it will never produce nuts. There have to be 2 trees of different genetic lineage in order for them to cross and make seeds. So they're somewhat limited in what they have out there.
How soon will they be available? How soon will these be available?
We are literally, I think, a week away from submitting our petition to the USDA, and once that goes through, the EPA will look at it. We have another petition to the FDA, and that will probably take about 2 years. So they're going to be soon. Soon is the answer. The pollen will be available first, rather than nuts. So as soon as, because we have a bunch of stored pollen, if you had an American chestnut tree with female flowers, we can mail you pollen, and you can pollinate those right up as soon as it's available. The seeds are going to be we have to distribute some of them to the New York State chapter, actually gets first dibs. So if you do join, there's a little thing in here where it's TACF New York chapter. If you're a member of the New York State American Chestnut, I think it's like either you get a seedling or you get seeds or you get pollen. First come, first serve. They get the majority of the materials that's first available since they've been funding us for the past 20 years or so. 30 years, sorry. More decades in there than I care to think about. So, Yes?
If you crossed two transgenic trees down the road sometime, what would be the expected success rate of their offspring?
Then you'd have, since it's on one chromosome each, you'd have 75% of the offspring have the gene, but then you would, our trees in our seed orchards right now are a little, we don't want to do that yet because they're a little too closely related. We want to avoid tree incest. So we're going to keep outcrossing, like we figure half is good for now, and as long as we keep getting more and more genetic diversity, at some point you could take a tree with a Georgia background plus the transgene and a tree from a, West Virginia background and cross them and get even, it would have 2 copies of the resistance gene in there, and then yeah, every offspring it had would then have resistance. But we don't want to do that yet. So, no. Any other questions?
So this problem, was it a virus or a bacteria that came
It's a
up?
fungus.
It was a fungus.
It's a fungus, yep. So there's other pathogens as well. You've got the ambrosia beetle, loves chestnuts, but you can control that a little easier with insecticides and things. Nothing is really super resistant to ambrosia beetle, but it's not a huge, it's more of a, it won't kill the tree outright, it'll just make it unhappy. And then we have Phytophthora in the south where the ground is wet. There's a soil, another oomycete, it's like a water fungus that attacks the roots and that kills them all the way dead. But that's only if you plant in like really moist, wet, warm areas. And chestnuts prefer a nice, well-drained slope anyway, so we're not too worried about Phytophthora for the moment, although it's beginning to get a problem. It's a problem in Georgia and North Carolina as it moves northward with the warmer weather. So
I'm curious about why does the chestnut grow 7 feet in a year's time? Are there other natural predators?
Deer love it.
Deer love it too.
We've seen deer jump like 10-foot fences to get into the chestnut orchards, and they'll just no, browse the top right off. So we think that's why it's almost so good at sprouting, is to overcome herbivory and just keep growing.
Interesting.
So you'll want to have like a wrap or some sort of, we put like a little cage, a little fence cage around the little baby guys until they get tall enough that the deer can't nip the leader, the top of the tree.
Did our local indigenous Onondaga Indians eat the leaves in a tough winter?
Oh, they absolutely ate the nuts. That was one of the reasons they think some of these forests were just so well kept and the trees were allowed to get so big. They do a light burn to get rid of some of the underbrush and then collect the nuts. In some places the nuts would be 6 to a foot deep, and some of these chestnut forests would just be just a carpet of food. And the leaves are apparently, folklore is that they're good for like upset stomach, I think, chestnut leaf tea and some other, it's very high in antioxidants and other things. So yeah, it was definitely part of their medicine and food cycle, so yeah. We're working with the Center for Indigenous Studies at ESF with the Haudenosaunee and seeing how they feel about this, whether or not they want to have them on their land, and so far they're thinking about it. It's under consideration whether or not they want to.
Is the chestnut flower edible?
The flower? I don't know. I haven't tried it. I'll have to try it next year and get back to you on that. So I have had the nuts. They're fantastic. I prefer them to the Chinese. They've got a lot more flavor. The Chinese nuts are very starchy and they're good for making like a flour out of, but the American chestnut nuts, they're great. I wish I could bring some. They're all research labeled and numbered in our fridges, but yeah.
If I said that the city of Cortland is planting ruby red chestnuts, what would your comments be on that?
What's a ruby red chestnut? Is that
That's your comment?
Yeah, I have not heard of that, so I'll have to look at
Those
that.
are about the only chestnut trees that we've been planting in the city over the last 15
Are
years.
those horse chestnut?
Probably.
Okay, horse chestnut is a different species. It's in a different grouping. It's the one that has like the palmate leaves, and it has
And you don't want to eat that flower.
no, you don't want to eat that. And they have the conkers. They have the smooth nuts as opposed to the spiky ones. Or no, I'm thinking a different one. They have the horse chestnut. They have like, it's almost like a globe, and it has little points off of it. Is that what
The ones we planted haven't gotten big enough. I don't think they have nuts. They have a red flower, which is very
Okay, yeah,
outstanding.
chestnuts don't have a red, almost looks like this. I haven't heard of a chestnut with a red catkin.
The ruby red chestnuts, that's what
Okay.
we planted. So
I think they're horse chestnuts. I think that's a different genus of tree. So those are good street trees. So
I have another question.
Yeah.
The fungus you said lives on oak trees as well. It's not a threat to the oak tree, though.
nope, it just hangs out.
Okay.
Yeah. So, all right. Well, thank you for having me.
Excellent.
Feel free to come up and